The nucleus is the control center of the cell. It is vital to the survival of an organism. If you remove the nucleus from any cell, death is unavoidable. An amoeba can survive for a period of time after its nucleus has been removed, but dies unless the nucleus of another amoeba is transplanted. The nucleus contains eukaryotic cells' genetic library. It contains most of the genes in the eukaryotic cell. All eukaryotic cells except erythrocytes have an organized nucleus. The nucleus is visible under the light microscope and it's either disc or oval shaped. The size of the nucleus varies according to the rate of metabolic activity in a cell. A highly active cell has a large active nucleus. Generally, a cell contains only one nucleus. However, paramecium, liver, and muscle cells have at least two nuclei. Nuclear membrane. The hereditary material of a cell is surrounded by a lipid bilayer known as the nuclear membrane. It is formed from an extension of the endoplasmic reticulum. The nuclear membrane is structurally similar to the plasma membrane but differs in that it has ribosomes on it and contains pores. These pores are involved in the transport of materials between the cytoplasm and nucleoplasm. In the prophase stage of the mitosis, the nuclear membrane disappears and the cytoplasm and nucleoplasm mix. Towards the end of the cell division, these structures are represented by the endoplasmic reticulum and Golgi. Nucleoplasm. The semifluid which fills the nucleus is termed as nucleoplasm. The viscosity of nucleoplasm is higher than cytoplasm. Nucleolus. The nucleolus is a small coiled structure within the nucleus. It contains DNA, RNA, and proteins, and it is site where RNA accumulates after its synthesis at the chromosomes. The RNA is combined with the histone protein and is moved to the cytoplasm to form ribosomes. It is visible with histone proteins and is due to its high light refracting properties. Also, the other structure of the nucleus are stained using a basic dye. The nucleus is stained using an acidic dye. A single nucleus may contain one or more nucleoli which disappear during cellular division. Hereditary material. The nucleus contains hereditary material in the form of DNA, chromatin, and chromosomes. The double-stranded helical structure of DNA is associated with chromatin. During cell division, DNA is wound around histones, made of proteins that behaves as a bobbin for a long DNA molecule. During metabolic activity, the DNA unwinds in order for genetic information to be transcribed.
Mitosis. Recall that all cells are formed by the division of pre-existing cells. When a cell divides, the information contained in the DNA must first be exactly duplicated and the copies and then transmitted to each daughter cell through a complex series of processes. Most cell division in the body cells of eukaryotic involve in a process called mitosis, which is kind of a cell division in which a parent cell is divided into two daughter cells. The genetic makeup of both parent and daughter cells identically. A parent human body cell, for instance, has the same number of chromosomes as both its daughter cells. The whole process of mitotic cell division is complete in approximately an hour, and the number of chromosomes remains fixed in all cells. Interphase. The cell is carrying out its normal life activities. The chromosomes became duplicated. G1 phase. The length of this phase varies from a few minutes, hours of days to a few weeks. According to the type of cell in the G1 phase, the protein required for cell division, ATP, and the other cell structures and synthesized. Spindle fibers are formed in this stage. C phase. Following the G1 phase, it occurs within 6 hours in normal cells. During the S phase, chromosomal DNA is replicated in identical copies of each of its two halves. of chromatids and assembled. For instance, a diploid cell containing 2 sets of 8 chromosomes of 60 chromatids replicates during the S phase to produce a total of 32 chromatids. Since the S phase initiates division, a cell cannot divide without passing through this phase. G2 phase. It follows the S phase and is replicated within 4 or 5 hours. The replicated DNA condenses, shortens, and is visible if stained. Nerve and the muscle cells are incapable of dividing and remain in a phase known as G0. Karyokinesis. Karyokinesis occurs in 4 distinct phases: prophase, metaphase, anaphase, telophase. I'll talk about prophase. In early prophase, the nuclear envelope and the nucleus begin to disappear. Long thread-like bodies of chromatin begin to shorten and condense as visible. Toward the end of prophase, chromosomes continue to shorten and thicken. Spindles from between the centrioles which have moved to the poles of the cell. Metaphase. The formation of spindle fibers is complete by the beginning of metaphase. Spindle fibers attach to the kinetochores of the chromosomes. Chromosomes line up along the equatorial plane of the cell. The metaphase chromosomes orient themselves on the equatorial plate and are clearly visible with a stain. Each metaphase chromosome is composed of 2 sister chromatids attached to the spindle fibers of opposite poles. The chromatids are now ready to separate. Anaphase. The unique feature of anaphase is chromatid separation. Chromatids separate at their telomeres, and one group of chromosomes moves toward each pole. The microtubules of the spindle shorten considerably, pulling the chromosomes towards the poles. The events of telophase are almost the reverse of those of prophase. This phase begins as chromosome movement ceases. A new nuclear membrane is formed at each pole which surrounds the daughter chromosomes. Each chromosome begins to uncoil, becoming less condensed. Chromatin fibers are formed and metabolic activities are initiated. Finally, a nucleus appears within each nucleus. When a karyokinesis or division of nuclear material is complete, the result of 2 identical nucleus. Cytokinesis. Cytokinesis is a division of cytoplasm which follows